Thank you for giving me this opportunity to talk about my work on the effects of bisphenol A on female reproduction in several generations of mice. What I'd like to do today is first give you some background on bisphenol A, or BPA, and some background on the ovary. Then I'll share with you our data on the effects of prenatal BPA exposure on the ovary and female reproductive system, and give you some data on particularly ovarian development and fertility. Then I'll share with you our data on transgenerational effects of prenatal BPA exposure on the ovary and female reproductive system, and then leave you with some conclusions. So first I'd like to explain what is bisphenol A. BPA, the structure is shown here, was originally synthesized in 1891 and intended for use as a chemical estrogen to help prevent miscarriage. However, at the same time, diethylstilbestrol, or DES, was developed and determined to be a more potent estrogen, and it was used instead of BPA. Now, BPA was never used pharmaceutically, but it became widely used by manufacturers of polycarbonate plastics. And why are we concerned about this? Well, one major reason we're concerned is because BPA is everywhere. It's in plastic food containers, microwavable trays, utensils, water bottles. It's a major component in the lining of canned fruits and vegetables. It's a component in dental sealant. It's present in dental appliances such as night guards and retainers. It's present in plastic baby bottles and in thermal receipts. Now, the reason we're concerned about all of this BPA in plastics is because BPA leaches out of plastics. And I'm showing here the structure of polycarbonate plastics, and this is showing the two BPA molecules that are held together by an ester bond. Now, this ester bond is fairly weak, and when it's exposed to heating such as microwaving or cleaning or aging or sunlight, that bond can break, causing the BPA to leach out of the plastic. Now, we're concerned about this because BPA enters the body, and we know that BPA is found in at least 95% of human urine samples, indicating nearly constant exposure to BPA. We also know that BPA can rapidly cross the placenta and enter fetal organs in animal models, and that BPA has been detected in the blood, serum, urine, liver, placenta, umbilical cord blood, and amniotic fluid in humans. And of interest to my laboratory, BPA has been detected in the ovary, particularly in follicular fluids. Now, we know from many previous studies that prenatal BPA exposure affects reproduction. Particularly, we know that it can cause altered cyclicity, impair ovulation in adult life, cause premature breast development, early puberty onset, and abnormal chromosomes and oocytes. What we were interested in, what other effects does prenatal exposure have on the ovary? Now, before I talk to you about that, I'd like to give you some brief background on the ovary. So the ovary has two major functions. The first is to ovulate eggs, or oocytes, and this is absolutely required for fertility. The second is to synthesize and secrete sex steroid hormones, such as estrogen and progesterone. And these hormones are critical for development of the eggs, implantation, menstrual estrous cyclicity, maintaining the entire female reproductive tract, fertility, and for female health of non-reproductive systems such as the cardiovascular, brain, and bones. Now, the way in which the ovary makes eggs and steroids is quite complex. And I'm showing you a schematic of folliculogenesis in the ovary. Now, what happens in prenatal life, when the ovary is developing, there are structures called germ cells nest. Now, these are germ cells that are tightly connected to each other and surrounded by a single layer of somatic cells. Now, at some point during prenatal life, some of the germ cells in the nest start to die, as indicated by the black dots here, by a process called apoptosis. This causes the nest to break apart, allowing individual germ cells to be present in the ovary and not tightly connected to each other. What then happens is the somatic cells that were surrounding the entire nest infiltrate and begin to surround individual germ cells. This results in the formation of what we call a primordial follicle pool. Now, primordial follicles consist of the oocyte, or the egg, surrounded by a single layer of somatic cells called granulosa cells. The important thing about these is that females are born with a finite number of primordial follicles. This is the full complement of germ cells that any female mammal will have throughout her lifetime. Now, these primordial follicles, they must grow into what we call primary follicles, then preantral follicles, and then antral follicles. Now, antral follicles are the only follicle capable of ovulating an egg, so they're absolutely required for fertility, and antral follicles are the primary producers of sex steroid hormones. So if you have a chemical exposure that affects this process, you can affect fertility and steroid production. Now, we were particularly interested in whether BPA affects the process of germ cell nest breakdown and whether it affects the ability of the ovary to form this finite pool of primordial follicles, because we thought that if something happens during this period, it could be setting up the animal for a lifetime of reproductive problems because they would not have a normal germ cell nest breakdown and formation of the primordial follicle pool. So the hypothesis that we tested was that prenatal low-dose BPA exposure affects germ cell nest breakdown, reducing the number of primordial follicles at birth. To test this hypothesis, we used the following experimental design. We dosed pregnant female mice from embryonic day 10.5 to birth with either tocopherol strip corn oil, which was our vehicle control, or BPA at 3 different doses, 0.5, 20, and 50 micrograms per kilogram per day. We selected this dosing period because this is the time when germ cells are forming nests and beginning to undergo germ cell nest breakdown. After birth, we collected ovaries on postnatal day 4 to do histological evaluation of germ cell nest and primordial follicle numbers. We also performed fertility tests on some of the animals at 3, 6, and 9 months of age. And I would like to mention that we selected the concentrations of BPA that we did because the 50 microgram per kilogram per day is thought to be the EPA recommended or safe dose of BPA. So we used that dose and 2 lower doses. So I'll first show you our data indicating that BPA inhibits germ cell nest breakdown in the F1 offspring. On the left, I've got a graph showing the number of germ cells that were remaining in germ cell nests in our control and 3 different treatment groups of BPA. And on the right, I've got the percentage of structures that are primordial follicles in our control and 3 different concentrations of BPA. What these data show is that BPA significantly increases the amount of germ cells that remain in nests and decrease the number of primordial follicles that form. This suggests that BPA is inhibiting germ cell nest breakdown because germ cells are staying in the nest and not going on to form primordial follicles. Now, I don't have time to show you all the data, but CQ Joe in the lab has done a number of experiments to look at the mechanism by which BPA is inhibiting germ cell nest breakdown. And what she found is that it does so by blocking apoptosis. So that normal process of germ cell nest breakdown requires some of the germ cells in the nest to undergo apoptosis to allow breaking of the nest apart and allow formation of primordial follicle pool. What CQ has shown is that BPA significantly increases the expression of anti-apoptotic factors and reduces the expression of pro-apoptotic factors. In addition, it decreases expression of FAS, which is a pro-apoptotic factor, and caspase-8, which is an executioner of apoptosis. And this is happening at postnatal day 1 as well as postnatal day 2, which then we think leads to a reduction in apoptosis on postnatal day 4 and a decrease in germ cell nest breakdown. Now, given these data, we wondered what are the consequences of this prenatal BPA exposure on fertility in later life. As I mentioned before, interference with formation of the primordial follicle pool can have consequences for fertility. So what we wanted to do was examine fertility in these animals, and we did so at 3, 6, and 9 months. So this is showing the effect of BPA on fertility in the F1 offspring at 3 months. In this table here, I've got the different treatment groups, the number of breeding pairs in each group, the number that had fertility problems, the number of pups per litter, and the number of dead pups. And what I'd like to point out that in the controls we did not have any animals with breeding problems, but our lowest dose and highest dose of BPA, we had some animals with fertility problems, and in the lowest dose of BPA we had significantly greater number of dead pups. When we looked at 6 months, we see that none of the controls had fertility problems. However, all 3 doses of BPA had breeding pairs with fertility problems. In the highest dose of BPA, we had significantly fewer number of pups per litter, and in all 3 doses of BPA, we had a significantly greater percentage of dead pups. And when we looked at 9 months, again, we see the animals here are getting a bit older, so we do have 2 in the controls that had breeding problems, and this is typical of aging mice. However, we had a greater percentage in the BPA treatment groups that had fertility problems. And in fact, in our lowest group, we had no live pups, and 100% of the pups were dead. And in the highest BPA group, we also had a significantly higher percentage of dead pups compared to the control. Now, we looked at different reasons for these fertility problems, and I put them here in tabular form. So we had some animals that had a fertility problem because of problems with ovulation. We had none of these in the control, but we had some in the lowest dose and middle dose of BPA. We also had some animals that had fertility problems because of problems in mid-gestation. They got pregnant, but they lost that pregnancy during mid-gestation. Our 2 control animals at 9 months had this problem, but we also had several in the low, middle, and high dose of BPA. And then finally, we had some animals that had a late gestation problem where we had none of the controls had this problem, but in the 3 different doses of BPA, we had animals that got pregnant and then lost their pregnancy in late gestation. Now, given these effects on germ cell nest breakdown and fertility, we wondered, were these effects transgenerational? Particularly, we wanted to know whether the F2 and F3 generations of mice had problems with germ cell nest breakdown and reduced female fertility. So I'm showing here first data that I've shown you before in the F1, indicating that BPA inhibits germ cell nest breakdown. And again, BPA significantly increased the number of germ cells in nests and decreased the number of primordial follicles. Now, when we look at this in the F2 generation, we did not see any significant differences between BPA and vehicle at the number of germ cells in the nest or in primordial follicles. And in the F3, again, we did not see any difference in numbers that remained in the nest versus primordial follicles in any of the treatment groups, suggesting that the effects of BPA on germ cell nest breakdown are not transgenerational. They occur only in the F1 and not the F2 and the F3. We then wanted to look at whether fertility issues were transgenerational. Now, these are data I've shown you before, but in tabular form, on the effect of BPA on fertility over time in the F1. So I've got here the percent of fertile females at 3, 6, and 9 months, and I want to point out that the controls, we have a slight decline as the animals get older, but with the BPA treatment groups, we have significant decline in fertility over time, and it's much greater than we see in the control. In fact, our lowest dose of BPA has a really profound effect on fertility over time compared to the control. When we look at this in the F2, we see a similar situation. So we see a very slight decline in fertility in the controls as they age, but however, all 3 doses of BPA significantly reduce fertility over time in the F2, and again, we see some of the most profound effects in our lowest dose as well as our middle dose. Similarly, when we look in the F3, we don't see dramatic differences between the control and the middle and high dose of BPA, but again, it's our lowest dose of BPA. We see a dramatic decline in fertility over time in the F3, suggesting that the effects of BPA on fertility over time are indeed transgenerational in nature. So to summarize, I've shown you that prenatal BPA exposure blocks germ cell nest breakdown in the F1 generation, and that it reduces female fertility in the F1, F2, and F3 generations. And this fertility problems are due to reduced ovulation, miscarriages, either in early or late gestation, and dead pups. And I've also shown you that these effects of BPA may get worse with age and persist in future generations. So in conclusion, prenatal exposure inhibits germ cell nest breakdown, but this effect of BPA is not transgenerational. And prenatal BPA exposure reduces fertility in a transgenerational manner. With that, I'd like to take a minute to acknowledge several collaborators on this work. I'd like to acknowledge Dr. Jay Coe and his research assistant Patrick Lynn, who are very involved in this study and studying the males from these experiments, and hopefully you'll be able to hear from him in the future about his data. I'd like to thank Dr. Pan from Nutritional and Food Sciences, who is going to help us look at whether our transgenerational effects are due to epigenetic changes. I'd like to thank Liang Gao, who's my lab manager and really helped organize these experiments and helped with many, many aspects. Dr. Rex Hess, who is helping Dr. Coe study the males in these studies. And I'd like to thank Drs. Eilert Zivgau and Wei Wong, who did the studies on the fertility as well as the F1 generation, and Amy Berger, who is an undergrad in the lab, who did all of the studies on the F2 and F3 generation germ cell nest breakdown. And I'd also like to thank some other wonderful students who are part of the center, Katie Chang, Shreya Patel, Emily Brown, C.K. Joe, and Sonia Rattan, who are not only working on this study, but working on phthalate studies. In particular, I would like to point out thanking CQ, because she is the one that did the studies to determine that germ cell nest breakdown is inhibited by BPA through a mechanism that involves inhibition of apoptosis. And finally, I would like to thank our funding sources and thank all of you for your attention, and I'd be happy to answer any questions.